Picture this. It's around 30,000 years ago, somewhere along the ancient Japanese coastline, and a small group has found a tidal pool absolutely loaded with fish that the receding tide has temporarily trapped in a shallow depression. The fish are right there, not running, not hiding, barely moving. One of the younger group members, driven by hunger and the kind of opportunistic thinking that has always characterized our species at its most practically creative, reaches in, grabs one, and eats it directly, raw, without fire, without any preparation, without any of the processing that most animal protein in their diet normally receives before consumption. The result is not immediate catastrophe. The result is, in fact, a perfectly edible meal. Nobody gets sick. The fish goes down without incident, provides nutrition, and the group moves on with their day with exactly the same level of collective drama that accompanies any other successful food acquisition, which is to say, not much. Now imagine the same scenario but with a deer carcass that's been sitting in the open for 12 hours. Same person, same hunger, same practical opportunism, same absence of fire nearby. The outcome of eating that raw meat without any processing is a dramatically different story that involves the kind of internal biological event that ruins several subsequent days and potentially shortens a life if the pathogen load happens to be high enough. So here's the question. This video exists to actually answer: why can humans eat raw fish without immediate catastrophic consequences in ways that raw terrestrial meat often doesn't allow? This sounds simple. And it is until you start examining why it's true, at which point it becomes considerably more interesting, more biologically, and more connected to ancient human dietary history than most people expect. Stick around because the answer crosses microbiology, food chemistry, evolutionary biology, and the fascinating history of how ancient human coastal populations developed eating practices that we still find reasonable today, and by the end, your relationship with the sushi menu will be slightly more informed and slightly more complicated. Begin with the most important clarification this topic requires, because the premise contains a significant qualification that needs stating directly. Humans cannot eat all raw fish safely any more than they can eat all raw meat safely. The statement that raw fish is safer than raw meat is a statistical and contextual observation rather than an absolute biological rule. And understanding where and why it's true requires understanding what makes raw animal protein dangerous in the first place. The relevant factors are pathogen type, pathogen load, the biology of the animal being consumed, the animal's environmental exposure, handling conditions after death, and the time elapsed since death. Raw fish in contexts is safer than raw terrestrial meat in most contexts for reasons rooted in all of these factors simultaneously. The pathogen categories that make raw meat dangerous divide into bacteria, parasites, and certain viruses, and each category behaves differently across fish versus terrestrial meat contexts in ways that explain the statistical safety difference. Start with bacteria, because bacterial contamination is the most immediate and most common concern with raw animal protein in any context. The bacteria most associated with dangerous foodborne illness from terrestrial meat, including Salmonella, E. coli, O157:H7, Campylobacter, and Listeria, are fundamentally warm environment organisms that thrive in the digestive systems, fecal matter, and environmental contexts of warm-blooded animals living in warm terrestrial environments. Fish live in cold water, and cold water bacteria are different organisms operating under different temperature constraints than the bacteria that dominate warm-blooded animal digestive systems. The core body temperature of a living fish matches the water temperature around it, which in most fish habitats is substantially colder than the 37 degrees Celsius internal temperature of a cow, pig, or chicken. The bacterial communities that colonize fish flesh are adapted to these colder temperatures and are generally different species from the warm environment bacteria that make raw chicken notoriously dangerous. Many of the bacteria naturally present in marine fish flesh don't grow effectively at human body temperature, which means that even if they survive consumption, they don't proliferate efficiently within the human body in the way that warm-adapted pathogens do. This temperature ecology connection is fundamental. A bacterium that optimizes its growth at 4 degrees Celsius is biologically quite different from one that optimizes at 37 degrees. And most of the dangerous human pathogens in the foodborne illness context are the latter type, warm temperature organisms that evolved in warm animal environments. Terrestrial livestock carry enormous populations of these warm adapted bacteria in their gut microbiomes. And when slaughter occurs without perfect hygiene, these bacteria contaminate meat surfaces and potentially interior tissues through the processing workflow. The average chicken at slaughter carries Campylobacter in its gut at concentrations that would concern anyone who understood what they were looking at, and processing inevitably transfers some of this contamination to the meat surface. Marine fish, by contrast, have gut microbiomes dominated by cold-adapted marine bacteria that are less dangerous to human health at body temperature. They're not sterile, and their flesh and gut contents absolutely do contain microorganisms, but the microorganism community profile is substantially less threatening to warm-blooded humans than the community profile in terrestrial livestock because the ecological conditions that produced it were so different. The cold marine environment doesn't favor the warm-adapted human pathogens that make terrestrial raw meat hazardous. The parasite situation is where the fish versus terrestrial meat comparison becomes more complicated and requires more careful examination, because parasites are actually more associated with fish in popular awareness, and the picture is more nuanced than the bacterial comparison. Raw fish does carry parasite risks, from Anisakis nematodes and various other fish parasites. And these risks are real and not trivial. Anisakis larvae in fish flesh can cause anisakiasis if consumed, producing gastrointestinal inflammation, pain, and sometimes serious allergic reactions in sensitized individuals. The risk is manageable through proper handling, including sufficient freezing at appropriate temperatures that kills the larvae, and the traditional Japanese practice of using extremely fresh fish for sashimi. Combined with experienced assessment of fish quality, reduced but didn't eliminate this risk. Terrestrial meat carries its own parasite risks, most famously trichinella in pork and various other species in game meat, along with Toxoplasma gondii, which is present in significant proportions of global livestock populations. The critical difference in the parasite comparison is that many fish parasites are visible to experienced handlers and can be detected and removed, and that the parasites commonly found in marine fish don't have humans as their primary or preferred host in the same way that some terrestrial meat parasites do. Trichinella in pork is adapted to mammalian hosts and survives and reproduces effectively in human tissue in ways that create serious infection. Anisakis larvae encounter the human gut as an accident of mistaken host identification and cause inflammation primarily through their attempts to penetrate gut tissue looking for a host environment they're not adapted for. The freshness variable is critically important and explains why the ancient coastal human who ate raw fish from a tidal pool was making a reasonable choice while someone eating raw meat from a carcass that had been sitting in warm conditions was making a poor one. Bacteria multiply rapidly on animal protein at warm temperatures. The doubling time for many dangerous bacteria under warm conditions is 20 minutes or less, meaning a moderate bacterial load on meat surfaces can become a very large bacterial load within a few hours of warm conditions. Marine fish held at near freezing temperatures in cold water until the moment of consumption maintained dramatically lower bacterial loads than terrestrial meat processed through warm ambient conditions even briefly. The ancient human eating raw fish from a tidal pool was consuming fish that had been alive moments before and was still being cooled by cold seawater. The total elapsed time since death was measured in minutes, and the bacterial load on flesh that had been cooled by cold water throughout the animal's life was minimal. The Saur, ancient human who might consider eating raw deer meat from yesterday's hunt, was dealing with meat that had been at ambient temperature, potentially quite warm, for many hours, with every surface exposed to environmental bacteria, insects, and cross-contamination from gut contents during processing. The difference in bacterial load between these two scenarios is vast and explains much of the practical safety differential that ancient coastal peoples would have experienced empirically without any understanding of the microbiology involved. The practical discovery of raw fish safety by ancient coastal humans represents exactly the kind of empirical learning through observation and outcome tracking that we've documented throughout this series as the mechanism for ancient human food knowledge accumulation. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. Ancient coastal peoples didn't understand bacteria or parasites. What they understood was that eating fresh fish from cold water was a reliable food strategy that didn't produce the illness outcomes that sometimes accompanied eating other raw animal protein. This empirical pattern accumulated across generations of observation and transmitted through oral culture produced culturally embedded practices around raw fish consumption in coastal ancient populations that have persisted in various forms into modern food culture. The Japanese culinary tradition of sashimi and the various other raw fish traditions across Pacific and coastal cultures worldwide represent the modern inheritance of this ancient empirical discovery, formalized into preparation practices, quality assessment criteria, and food culture that encodes the ancient learning in cultural form. A skilled sashimi chef assessing fish quality before serving it raw is applying accumulated cultural expertise that traces back through a very long chain of transmitted knowledge to ancient coastal people learning which fish in which conditions were safe to eat raw. The chemical composition of fish flesh versus terrestrial meat contributes additional factors to the safety comparison worth examining. Fish muscle is structurally different from mammalian muscle in ways that affect both its palatability and its safety when consumed raw. Fish muscle fibers are shorter and more loosely arranged than mammalian muscle fibers, with connective tissue that is less dense and collagen that is more thermally labile. This structure makes raw fish muscle naturally tender in ways that raw beef or pork are not, which contributes to the culinary palatability of raw fish that makes traditional preparations appealing rather than merely tolerable. The fat composition of fish flesh differs substantially from terrestrial mammalian fat, with fish fats being predominantly unsaturated and particularly rich in omega-3 fatty acids that are both nutritionally valuable and chemically distinct from the saturated animal fats that dominate terrestrial meat. Omega-3 fatty acids in fish flesh, while nutritionally excellent, are chemically reactive and oxidize more rapidly than saturated fats, which is part of why fish quality deteriorates more quickly than terrestrial meat and why freshness is so critical to safe raw fish consumption. The chemistry that makes fish fat nutritionally valuable is the same chemistry that makes fresh fish fat smell like the ocean, and old fish fat smell like something quite different and considerably less appetizing. The practice of using acid treatments, most prominently lime or citrus juice, in preparations like ceviche from South American coastal cultures demonstrates another traditional approach to raw fish safety that operates through chemistry rather than heat. The acid environment created by citrus juice denatures proteins in fish flesh, creating a texture change that looks and functions somewhat like cooking despite involving no heat, and simultaneously creates conditions hostile to bacterial survival. This technique, developed by ancient coastal South American peoples and refined across generations of practice, is another example of ancient human food technology discovering through empirical trial and outcome observation, a solution that modern food science can explain mechanistically after the fact. The role of salt in traditional raw fish preparations like gravlax and various fermented fish traditions represents a different chemical intervention with similar bacterial management properties. Salt creates osmotic conditions that are hostile to bacterial growth, reducing moisture availability in fish flesh to levels that significantly inhibit bacterial proliferation. Ancient preservation techniques using salt for fish, documented across multiple coastal cultures in different parts of the world, extended the window of safe fish consumption beyond what fresh fish alone allowed, while often producing flavor profiles through controlled enzymatic and bacterial activity that became valued food products rather than simply preserved raw fish. The fermented fish traditions across various Asian cultures, including certain preparations in Scandinavia, represent the extreme end of this spectrum, where controlled microbial activity transforms raw fish into products with safety characteristics quite different from fresh raw fish. Through the development of bacterial communities that produce acidic environments hostile to pathogenic species while contributing flavor compounds that characterize the fermented product. These traditions, some of which produce results that are challenging for uninitiated palates, represent sophisticated ancient food technology developed through the same accumulate and transmit knowledge process that produced every other ancient food preparation technique this series has examined. The question of why raw terrestrial meat is more dangerous than raw fish has a parallel answer in the question of why cooking made such a dramatic difference to terrestrial meat safety and somewhat less dramatic difference to fish safety in terms of pathogen load reduction. Heat kills bacteria and parasites with reliability and efficiency, and cooking large terrestrial animals eliminated the bacterial loads and parasites that accumulated in warm-bodied, warm-environment animals living in proximity to their own waste and to soil contaminated with pathogen sources. The transformation of human dietary safety that accompanied reliable fire use, which we covered in detail in our fire video, was partly a transformation relevant to terrestrial meat safety. Fish cooking produces genuine safety improvements as well, particularly regarding parasite elimination. But the baseline bacterial challenge with fresh fish from cold water was lower enough that raw consumption remained a viable strategy in ways that raw terrestrial meat consumption was not for most of ancient human history. Coastal peoples with consistent access to fresh cold water fish had a reliable raw protein source that didn't require fire for safe consumption in the way that terrestrial meat did, which may have contributed to the dietary patterns and the food cultural practices around raw fish consumption that distinguish coastal ancient human populations from inland ones. Ancient human populations that relied heavily on marine resources, a group that includes many of the ancient coastal populations whose archaeological record we have the richest evidence for, likely maintained immune systems and gut microbiomes shaped by continuous exposure to the microorganism communities associated with marine food sources. This continuous calibration of the immune and digestive systems toward marine pathogen communities is the same mechanism we discussed in our dirty water video regarding how continuous environmental exposure shapes immune function, applied to the dietary pathogen context. Ancient coastal humans eating raw fish regularly across lifetimes would have had immune systems well acquainted with the organisms in marine fish flesh, providing additional resistance to the manageable pathogen loads present there. The contrast with inland ancient humans who occasionally accessed fish but primarily consumed terrestrial protein is instructive. An inland ancient human who infrequently ate raw fish would have had less immune familiarity with marine fish pathogens and less gut microbiome adaptation to marine food sources, making even relatively low marine pathogen loads more potentially problematic than for consistently coastal populations. This contextual difference in safety reflects the general principle, discussed across multiple videos in this series, that ancient human food safety was partly about accumulated immune calibration from consistent exposure, not just about the inherent properties of the food itself. The modern context adds important layers to this ancient story, because modern industrial food production has substantially changed the pathogen dynamics of both fish and terrestrial meat in ways that partially alter, but don't, fundamentally reverse the ancient safety comparison. Modern commercial fish, transported at cold chain temperatures and processed under hygienic conditions, maintains much of the baseline safety advantage of cold water origin. Modern commercial raw fish safety is further enhanced by regulatory requirements for freezing at temperatures that eliminate viable parasite larvae, making modern sashimi-grade fish both commercially standardized and considerably safer than what ancient coastal populations were eating in terms of parasite risk. Modern commercial terrestrial meat production has introduced pathogen management at scale through temperature control, hygienic processing, and pathogen reduction interventions, but the fundamental warm animal, Warm environment pathogen community that makes raw terrestrial meat risky persists and continues to make raw commercial beef, chicken, and pork genuinely dangerous without proper cooking for most consumers without specialized tolerance from extensive raw meat exposure. The notable exception in the modern culinary world is beef tartare and similar preparations, which use very fresh, high quality beef handled with care to minimize surface contamination and consumed immediately, exploiting the same freshness and handling principles that make raw fish safer, applied to the most surface contamination prone of the terrestrial meats with strict quality controls. The comparison between high quality fresh beef prepared as tartare and sashimi grade raw fish illustrates that the safety difference between raw fish and raw terrestrial meat is not absolute, but contextual and probabilistic. Under ideal freshness and handling conditions, high-quality beef can be consumed raw with manageable risk, as global culinary traditions including steak tartare, Korean yuko, and various other raw beef preparations demonstrate. But the baseline probability of safe raw consumption without quality protocols and freshness guarantees is substantially lower for terrestrial meat than for marine fish in cold water, because the biological systems and pathogen communities involved are genuinely different. Ancient human populations discovering these differences empirically were making rational, experience-based dietary decisions without mechanistic understanding of why their observations were accurate. The coastal population that developed reliable raw fish consumption practices was not making a culturally arbitrary choice about food preparation. It was following accumulated empirical evidence that this food preparation approach was reliably safe in ways that comparable approaches with terrestrial protein were not, and encoding that evidence in cultural practices that transmitted the knowledge across generations without requiring each generation to rediscover it through personal illness outcomes. This is again the fundamental pattern of ancient human food knowledge that recurs throughout this series. Accurate practical knowledge preceding mechanistic understanding by thousands of years, transmitted through cultural practices and oral tradition, in forms that preserved the practical truth without requiring the scientific explanation. A traditional Japanese fish market worker in ancient times who could assess fish freshness by smell, appearance, and texture with extraordinary accuracy was applying accumulated cultural expertise that traced back to ancient coastal populations, learning what safe raw fish looked and smelled like through exactly the outcomes-based observation process that characterized ancient human knowledge development across every other domain. The bottom line that this entire examination supports is that raw fish and raw terrestrial meat are not symmetrical food risks, and the reasons they're not symmetrical are rooted in the biology of the animals, the microbiology of their environments, the chemistry of their flesh, and the practical parameters of freshness and temperature that determine pathogen load at the moment of consumption. Ancient coastal humans who ate raw fish were not taking equivalent risks to ancient inland humans who might have considered eating raw terrestrial meat, and the different food practices around raw fish versus raw terrestrial meat that developed in different ancient human populations reflect genuine empirical differences in safety outcomes rather than arbitrary cultural divergence. If this kind of investigation into the food chemistry, microbiology, and ancient human dietary practice underlying modern food safety intuitions is your thing, there's considerably more territory worth exploring in this direction. How different ancient food preparation traditions developed as empirical solutions to food safety challenges without mechanistic understanding of the problems they were solving, what the immune and gut microbiome differences between populations with different raw food consumption histories tell us about how dietary exposure shapes physiological tolerance, and what modern food science has revealed about the ancient preparation techniques that turns out to confirm their empirical logic with mechanistic explanations that ancient practitioners would have found both vindicating and entirely baffling. So stay around, because the biology of what ancient humans ate and why they prepared it the way they did is considerably more sophisticated and more interesting than simple necessity would suggest.